Hi everyone. Today we will be starting with a new chapter which is Biomolecules. This is the third chapter for your unit cell and it has a lot of weightage if you consider NEET, fine, which you might have already noticed. So if you think about this chapter only, there are at least four questions asked. Okay, and if you combine biomolecules, which you have studied in chemistry as well as biology, then the whole topic has at least six, okay, 5 to 6 questions asked from this only chapter. Okay, if you combine chemistry as well as biology, so it shows you how much important it is. If you just do this one chapter properly, you can get 20 to 24 marks, okay, just from a single chapter. Also, it is comparatively very simple now for you at least, right? So without much ado, let us begin. What are biomolecules, or what is this name? So actually, biomolecules are you study them in a subject called biochemistry, which is nothing but a study of all the chemical reactions and chemical molecules inside living system. So for example, let us just take what you see in your own house, right? You have let's say a tulsi plant at least, or any plant you have. Then there may be ants or cockroaches or some other living organism apart from you. If I tell you that both you and that ant are made up of the same kind of molecules, right? So at the molecular level, everybody is made up of the same kind of molecules, and they are called biomolecules. Fine. So let us just analyze this. We can do analysis by taking liver, piece of liver. Okay, this liver is nothing but an example of animal tissue. You can take any tissue you want to find out, okay, its composition or analyze. Then from a plant tissue, we can take a leaf. And for microorganisms, you can take a bacterial culture. Now, if you do not know what bacterial culture is, let me tell you. Can you see here a test tube, like, right? A test tube which is covered with something. Okay. So let me just tell you what it is made up or what this bacterial culture means. It is basically nothing but a liquid. Okay. It is made up of water, and along with water there will be minerals, there will be vitamins. Then for source of energy, okay, or source of food, there will also be some form of sugar. Okay, it could be glucose, fructose, sucrose, anything. And for growth, it requires proteins and amino acids. So all of those things we will dissolve in the water, and finally what you get is called media. Okay, remember this, media. In this nutrition rich media, when you add bacteria, the bacteria will take up all the nutrition and it will grow. So that growing bacteria in this nutritional media is called a bacterial culture. Why it is covered? To make sure that no other bacteria or fungus can fall inside and utilize this nutritional media. Right? So like this, you can take bacterial culture of, let us say, E. coli, or if you take a common thing from your house, it would be chash, right? If you take buttermilk and analyze it, you will find the same kind of things, fine? So an animal tissue, a plant tissue, or a microorganism, you can take any one as a sample. We will continue understanding using a liver piece. So now what you do with this liver is you grind it, okay, with a mortar and pestle. This is a mortar and pestle. You put the piece of liver there, add something, like it's just like making chutney. But while making chutney, you have to add water, right? So instead of water, you add here trichloroacetic acid. This trichloroacetic acid will help you in grinding the liver piece. Okay. Now, the name, I hope you know chemistry very well, right? Even if you don't, do not worry. Just keep trying. Fine. So trichloro. So three chlorines. One, two, three. Right? It has some molecule which has three chlorines. Trichloro. Acetic acid. Acetic acid is nothing but CH3COOH. Now here This trichloro means that the hydrogen have been replaced with chlorine. So you have 1, 2, 3, 3 chlorine, trichloro, acetic, and this COOH acid. Clear? So using this trichloroacetic acid, you have made a nice paste of the liver tissue. Now you take something called a cheesecloth or muslin, if you know. Okay, it is why it is called cheesecloth is because cheeses are made in such kind of cloth. Do you know how paneer is made? And ask your mother; she will remove all the excess water, okay, from the solid substance. So to remove or to separate solid from the liquid portion, you can use such type of a filter. Okay, such type of a cloth, which is called a cheesecloth. So you pass this paste through the cheesecloth, and this is the kind of thing you get. Okay, you will get something at the bottom, which will be trichloroacetic acid, plus whatever has dissolved from this liver tissue will also be present there. Whatever could not dissolve or was larger in size will be found up here, right? Okay. So whatever is remaining in your cheesecloth is called retinate. Okay, retinate, which is acid insoluble. It was not able to get solubilized in this trichloroacetic acid, so it remained above. So this is retinate. It is the acid insoluble fraction, and the bottom one is called filtrate. Okay, filtrate is the acid soluble part. Here you will find all the molecules which were soluble in the trichloroacetic acid and which were not soluble will be remaining up here. When you look at this filtrate, okay, when you do the chemical analysis, you find it is composed of a lot of organic compounds. Okay, many organic compounds are present in this filtrate. Okay, now what are these compounds? What are these chemical compounds, organic compounds? From chemistry point of view, we can identify them using their functional groups. Okay, if you say chemically what are these molecules, you can use the functional group to name them, right? So you can have aldehydes, CHO, ketone, C double bond O, ester, COO, carboxylic acid, COOH, or amides, and etc. etc. How many ever functional groups you have studied in chemistry, all of those can be included. But from a biological point of view, if somebody asks you that what are the organic molecules, organic biomolecules that you found in the filtrate, the answer will be amino acids, nucleotide bases like purines and pyrimidine. These are nothing but these are bases, right? Nucleotide base. You can have fatty acids, sugars like glucose, etc. A lot of things. So this is what we will be studying in this chapter. Amino acids, nucleotides, fatty acids, sugars, and a lot more. So now how do you exactly find out what compounds are present in this filtrate? So you take the organic compounds or that filtrate, then there is separation or isolation carried out. This separation can be done using many different chemical methods, and you separate the organic compounds. Okay, after separation or isolation, you identify that what has been separated, after which it can be purified to get only that single compound you want to study. And after purification, after getting only that one single compound, you can perform analysis to find out its molecular formula and its probable structure. For example, let us take methane. Let us say you identified, separated, and purified methane. After purification, you find out its molecular formula is CH4. And using that, you can even calculate or consider its structure, how it would be arranged. So this here is the tetrahedral structure of methane. Carbon, three hydrogens, one coming out of the plane, which is why it is darker in color, and one going outside the plane, okay, into the plane, which is shown in this dotted line. So this is how one by one all of these organic compounds are studied. Now that we know what compounds, organic compounds are or how they are analyzed, it has been found that the retinate, which is the upper portion, has something called biomacromolecules, and the filtrate, which is this lower portion, okay, acid soluble fraction, it has biomicromolecules. So we will study biomacro and biomicromolecules. What are the differences? Micro, wherever you hear the word micro, it means small, extremely small. Okay, so biomicromolecules are small molecules found in filtrate, and they are found in the acid soluble fraction. They are soluble in the trichloroacetic acid. The molecular weight is between 18 to 800 Dalton. Now your homework for today is to find out how much grams is 1 Dalton. Okay, 1 Dalton, 1 dA or 1 capital D is equal to how many grams? Okay, it is also sometimes referred to as atomic mass unit, AMU, or written just as U. So find out, that will be your homework. Okay, right, coming back to the point, biomicromolecules can weigh from 18 to 800 Dalton. Okay, I've shown it here itself. Never mind, 1 Dalton is equal to 1.6 into 10 raised to minus 24 grams. Now when we talk about the biomacromolecules, they are the larger molecules which are found in the retinate. Retinate is the acid insoluble fraction, whatever was remaining in the cloth. Its molecular weight is above 10 kilodalton. Okay, kilo means 1000, so more than 10,000 dalton. Its weight is, the exceptions are lipids. Lipids are normally, they have lower molecular weight compared to the other biomacromolecules. Biomicromolecules includes all the monomers to oligomers. Oligo, wherever you hear, it means few. Okay, so monomers, and when few monomers come together, they form oligomers. So biomicromolecules has monomer and oligomer. Biomacromolecule include proteins, which are polymers of amino acid, polysaccharides, which are polymers of sugar. Okay, and this here is glucose sugar, and when you make a huge polymer out of it, okay, it is called starch, which is a biomacromolecule. Nucleic acids, which are polymer of nucleotides, and then you have lipids. Okay, so from this, now you know that all organic compounds obtained from living tissue can be called biomolecule. Okay, now apart from the organic compounds, you will also find some inorganic compounds in the filtrate, right? So living organisms also have inorganic elements and compounds in them. Here, when you take the liver, again we start with the same piece of liver. You weigh it, and when you do the weight, this is called wet weight. Okay, wet weight because it still has water, it is still moist, it is fresh. You take the wet weight, then you dry it. You allow it to dry, it will become dehydrated. This means whatever was water weight, okay, whatever water was stored in this tissue will all be dried off. So now whatever is remaining in this tissue will be called its dry weight. Okay, this is weight without the weight of water. Clear? But now this tissue, it will have the inorganic elements as well as organic compounds. So what to do? We just want to find out what inorganic elements are present. For that, you burn this dried piece of liver. Okay, you burn this piece of liver. Whenever you burn something, the organic compounds, right, they get converted into carbon dioxide and water. Combustion. So after burning, when all the carbon dioxide and extra water has evaporated, what you are left with is called ash. Ash here, or whatever the powdery gray colored thing you get, it is the portion of that living tissue which will be having the inorganic elements. It includes sodium, potassium, calcium, magnesium, water, and some other compounds as well. Okay, like sodium chloride, calcium carbonate, phosphate, sulfide, sulfates, okay, etc. A lot of things. So this is how the inorganic chemical analysis is done of any living tissue. I hope it is clear till here. So again, when we started with the cell, we had discussed this proportion, right? Coming back to the same, if you see the composition of cells, average composition, they are made up of 70 to 90% water, followed by 10 to 15% proteins. Then nucleic acids, it includes DNA, RNA, okay? And then carbohydrates, followed by lipids, and the lowermost or the least amount will be of ions. All that sodium, potassium, chloride, sulfate, all of that will be included in the ion. Yeah, so this is the composition of an average cell. Now, what separates living from non-living? If I give you two samples, okay, I just give you ash from two different samples, how will you be able to say whether it had or it was organic, it was a living thing, or I just gave you some non-living thing? Fine. So that is when you compare living and non-living, you find out there is a huge difference. For example, here is a table of comparison given between non-living and living. In non-living, they have taken Earth's crust. Okay, the outermost layer of soil is Earth's crust, and they have taken composition of a human body. Fine. To start with the element hydrogen. So hydrogen in Earth's crust is just zero point 0.1, while in human body it is 0.5. You can see the difference, right? This will be true for all the living system. Okay, they will have higher hydrogen than higher carbon. Earth's crust has only 0.03% carbon, while the human body, it has 18.5%. Right? Life or living matter is organic in nature, which means it is made up of carbon, hydrogen, and oxygen, which is why you see these values here. Right? Oxygen followed by this. In Earth's crust, you have 46%, and in human body, 65%. Nitrogen is higher in atmosphere, but it is low in the Earth's crust. So there is very little nitrogen in the Earth's crust, but it is quite high compared to that in the human body. It is 3.3%. Then sulfur is also again less in the Earth's crust, more in the human body. Sodium is opposite. Okay, sodium is the only element or only ion which is not essential for living system. Okay, remember this word. It is not essential. You do not need to take sodium every day. Okay, even if you do not eat salt, if you do not eat NaCl, it will be okay. So sodium is higher in the Earth's crust, but it is not essential for human body, and it is only 0.2% of the human body. Then calcium higher in the soil, less in human body. Magnesium higher in the soil, less in the human body, followed by silicon, which is 27.7. Silicon along with oxygen makes silicon dioxide, which is sand, right? So sand is of course present more in Earth's crust, okay, and very negligible, very less in the human body, which is why you see this difference. So even without labeling, if I give you two samples and I give you this table, you should be able to say that which one is the living system or the living matter and which one is the non-living matter. Clear? Fine. Now, after having this common discussion, some logical and some chemical analysis based revision. See, sometimes they do ask questions based on this, okay? They will test your knowledge, they will test your understanding, which is why it was important to consider. Remember biomicromolecules, macromolecules, then what will be found in the filtrate and what will be in the retinate, okay? And uh, what all kind of analysis you can do, organic and inorganic, how you do it. Clear? Very good. Chalo. We begin with the first class of biomolecules, which are the carbohydrates. When you hear the name, carbo comes from carbon and hydrate. You know, like people tell you, stay hydrated, it is summer, keep on drinking water. So Hydrate actually means H2O, water. So, literally, the name carbohydrates comes from hydrates of carbon. Okay, so carbon, which is having some water, okay, H2O, in some way attached to it. Okay, so hydrates of carbon or carbohydrates. In common terminology, for people who exercise or who are very diet conscious, you would have heard the term carbs, right? So these carbs are nothing but your carbohydrates, the short form of carbohydrate. Right, let us begin. The major components of carbohydrates are carbon, hydrogen, and oxygen. Right, carbohydrate. They have variety of roles. What is the role of carbohydrate? Then they have a main role in energy metabolism. That is, they provide you with energy. Just think of glucose or sucrose, right? It is providing you with energy. So the carbohydrates, they have major role in energy metabolism. Okay, then energy storage. Think of a potato or radish or carrot. It is storing its energy in form of carbohydrate, right? In form of starch. So for energy storage also you use carbohydrates. For structural support, think of cellulose in when we discuss cell wall. Okay, so cellulose is again nothing but carbohydrate. It is not used in metabolism or storage of energy. But where else is it used? It is used for structural support. It provides that structural support to the cell, right? That rigidity, shape will be maintained because of this cell wall, which is mostly made up of carbohydrates. Previously, their empirical formula. I hope you know what empirical formula means. Empirical formula is your normal formula which you have shortened, okay, to make it easier for you to understand. So the empirical formula or the common formula was considered to be CnH2ON. Okay, Cn, N here can be any number, and then H2ON. Fine, but not all molecules follow this formula, and even if they do follow this formula, then not all of the molecules which show this CnH2ON is a carbohydrate. Okay, which is why this is no longer used. We do not use this. But just for your understanding, I am telling you that previously it was thought that the empirical formula was CnH2ON, but not all molecules which follow this formula are carbohydrates. And also, all carbohydrates do not follow this formula. Clear? Now these carbohydrates, they are classified based on the monomer, okay, or on the basis of the mers. You have monosaccharide. Mono means one. Saccharide comes from Greek word saccharon, okay, or saccharose, which means sugar, sweet. So monosaccharide means one, okay, here it is a monomer. Then disaccharide, di means two. Oligosaccharide means few, and polysaccharide, which means a lot. Always remember polysaccharide like polymer. Okay, right. So let us begin with the first one, which is a monosaccharide. Okay, it is the smallest molecule possible, which can be called a carbohydrate. Okay, it is the smallest molecule you can call a carbohydrate. The simplest form of carbohydrate, generally known as sugars. When you talk about sugar, you are mostly talking about monosaccharides, and we shall see later, or of disaccharides. Okay, they are polyhydroxyaldehydes or ketones. Okay, what do you understand by this? Poly means many, hydroxy is OH group. Okay, alcohol group, OH. So these molecules, these monosaccharides or carbohydrates, have many molecules of alcohol, okay, polyhydroxy, many molecules of OH, along with aldehyde, CHO, or ketone. Clear? So this is what a carbohydrate basically is. It is a polyhydroxyaldehyde or ketone. Monosaccharides do follow this formula, CnH2ON. Okay, the formula we saw previously is followed by the mono. saccharide. And just to make sure you understand this, just put 6 here. Okay, so C6 H2O whole 6. When you open the brackets, you get C6 H12O6, which is nothing but the formula for your glucose, right? So this is why it is said that monosaccharides normally follow this CnH2ON formula. Now, some properties of these monosaccharides. How will you identify that it is a monosaccharide? They are mostly colorless. Okay, colorless in the sense, in chemical sense, you always called white substance is colorless. Okay, so here what you see is your table sugar, which is colorless, odorless. Odor meaning smell. There is no smell of monosaccharide. Think about glucon D. If you have tried, it is nothing but glucose, right? So glucon D is white in color. You call that colorless. It does not have any smell unless it is some flavored. Okay, if it has some lemon or orange flavor, it will have some odor. Otherwise, the original glucon D does not have any flavor. Then it is crystalline. Like salt, you will find crystals of sugar, right? Crystals of monosaccharide. They are sweet in taste. When you eat glucon D, it is sweet in taste, right? And they are water soluble. You take one spoonful of glucon D and add it to water and mix it. It will dissolve in water, which means it is water soluble. And finally, it is optically active. Optically active meaning it can change the direction of plain polarized light. Okay? In chemistry, you must have studied this in detail. When you apply any plain polarized light on a sugar, it will either rotate it to the left or it can rotate it to the right. Some sugars we will see are not optically active, okay, only one or two, which are mostly trioses, but otherwise all of them are optically active. Now, how do you classify these monosaccharides? You classify them on the number of carbons, okay, on basis of number of carbons. It could be 3, 4, 5, 6, 7. Or, and you classify them on the basis of the functional group. The functional group can either be aldehyde or ketone. Now, you might have already studied or you must already know that aldehyde is CHO and ketone is C double bond O. Okay, they both the carbons here are called the carbonyl carbon and they are functional group isomers. Aldehyde and ketones are functional group isomers. When you study different kinds of isomers, you may have already also studied this type, right? So functional group isomer, one more example to add is aldehyde and ketone. Fine. Okay. Now let us start with the 3. Okay. The minimum amount of carbons required to make a monosaccharide is 3. Below 3, there is no monosaccharide. Okay. There are no carbohydrates. Carbohydrates start with 3 carbons minimum. These 3 carbon carbohydrate, if they have aldehyde group, they are called aldotriose. If they have ketone, they are called ketotriose. For 4, you can call them aldotetrose or ketotetrose. 5 is pentose, so aldopentose or ketopentose. 6 is hexose, so aldohexose or ketohexose. And 7 is heptose, okay? Heptose, so aldoheptose in case of aldehyde and ketoheptose in case of ketone. So this is how basically you classify the monosaccharides on the basis of carbons. The smallest monosaccharide or the smallest carbohydrate is made up of 3 carbons and it is called a triose. Triose can have aldehyde or ketone group and then you call it either aldotriose or ketotriose. Clear till here? Very good. So now let us consider few of these, okay? Just some examples we will see. Trioses have 3 carbons, so they are 3 carbon sugars. Putting that 3 in the formula CnH2ON. You put 3, so you get C3H2O3 will give you H6O3. Okay, so C3H6O3 is the formula, molecular formula for trioses. Clear? Example of trioses are glyceraldehyde and dihydroxyacetone. I know you would have heard of these terms, okay? Glyceraldehyde 3-phosphate, dihydroxyacetone phosphate in glycolysis and all, like glucose metabolism. You might have heard of these terms. So let us see what these molecules actually are or how they look like. How will you draw it? So here you have glyceraldehyde. We start by drawing a vertical line. You draw a straight line, and then based on the number of carbons, each end will be one carbon. Okay, on this vertical line, each end will be one carbon. So you have one carbon here, one carbon at the intersection, and the third carbon here. It is a 3-carbon sugar. So you have 1, 2, 3, 3 carbons, 1, 2, and 3. Now I'm telling you, and even it is written here, glyceraldehyde, aldehyde, which means one of the carbon is CHO. So always put aldehyde group or ketone group at the highest possible position. So you put aldehyde at the first carbon position and ketones at second carbon, always. So here you have aldehyde at first position, then at second position you just have one carbon, and at the third position always write CH2OH. Okay, at the last carbon you can, without looking at it, write CH2OH. Clear? So here I have told you this much. It is a triose sugar, which means it has 3 carbons. You can draw it like this: 1, 2, and 3 carbon. It is an aldotriose, so you can put CHO here and CH2OH on the third carbon. Now what is left to do is just add one OH and one H, and your glyceraldehyde molecule is ready. Right? Can you see how easy it is? Just draw a vertical line, draw 3 carbon atoms, and on this line, horizontal line, you have OH and H. Aldehyde will be given the number 1, and on the last carbon you can write CH2OH. Now let us make this diagram of dihydroxyacetone. Okay, dihydroxy, which means it has 2 OH molecules, and acetone is this. Clear? Let us see now. So this is also a ketotriose sugar. Ketone will always occupy position number 2. Okay, carbon will always be carbon number 2. So here you have CH2OH. Here the second carbon is the carbonyl carbon, C double bond O, and the last carbon is again CH2OH. As I had already told you, the end carbons you can write directly CH2OH. So here also both the ends are written as CH2OH. Now can you see dihydroxyacetone? Clear? And one means ketone, right? From the name itself you can come to know acetone or one is the suffix for ketones. So this is how you make the triose sugars. Fine, you have this 3 carbon sugar which is aldo triose, okay, glyceraldehyde, and ketotriose example is DHA. Now let us take example of hexoses, which are another famous sugars. Okay, they have 6 carbon, so the formula will be C6H12O6. It includes glucose, galactose, fructose, etc. There are about 8 or more 6 carbon sugars, okay, 6 carbon hexoses. But for your information, we will only be studying a few, which includes glucose, galactose, and fructose. Glucose is an aldehyde sugar. So draw with me. Now again, I know you do not have to appear for theory exam, but why this is important is you will be able to better understand the metabolism and what is actually happening when we study further chapters. Okay, so just for a sound logic and understanding, we have to study this. So you draw a straight line, the ends will be one carbon, right? Here there will be one carbon, and on this end also there will be one carbon. So you require 4 vertical lines. You draw the 4 vertical lines, put carbon everywhere. First position will be aldehyde, last position will be CH2OH. Now you just have to put OH and H on every side. You cannot haphazardly put it anywhere. Okay, you cannot put it anywhere. You have to follow um, a specific sequence. For glucose, I will tell you how I remember. When you say it, okay, I say it like this: OHH, OH, OH. Fine. When you repeat it 4 5 times, it will get stuck in your head. So now you know on the right hand side, you always have OHH, OH, OH. On the left, you just have to put H wherever there was OH and OH wherever there was H. Clear? And voila, you have this glucose molecule ready. You see how simple it is, right? Now, fructose is again a hexose sugar, but it is ketone. Okay, it has ketone functional group. So you do the same thing. You draw a vertical line, you draw horizontal lines, and now the ketone group will always be on the second position. So C double bond O. On the ends, you just write down CH2OH, CH2OH, and then whatever you were following in glucose, okay, this HOHOH will be seen here as well. HOHOH, and here you have fructose. See how simple. So this is how you draw the structures, or you understand the structures of monosaccharides. Clear? Now, something about configuration. Fine. The lowest asymmetric carbon. I hope you know what asymmetric carbon or chiral carbon is, right? Let us just consider in this previous one only. Let us see this glyceraldehyde molecule. This carbon, the central carbon, it has aldehyde group on one valency, OH or hydroxyl group on another, CH2OH, okay, on one side and H on the other side. The carbon which has four different functional groups or four different groups present on each of its valencies is called a chiral carbon. Okay, so this chiral carbon shows optical isomerism. Okay, it will be optically active. How will you find out which type of isomer it is? So if you write the last chiral carbon or the last asymmetric carbon, if it has the functional group OH or hydroxyl group on the right hand side, you call them dextrorotatory or D isomer, capital D. Okay, so here this will be the D-glyceraldehyde. If you write OH on the left hand side, it becomes L-glyceraldehyde. Clear? So D-glyceraldehyde, then here you have D-glucose. See, this is the same OHH, OH, and OH here, the last OH is on right hand side, so you call it D-glucose. If it is on the left, you call it L-glucose. Same way for ribose, which is a 5-carbon sugar. If it is on the left, you call it L-ribose. And here is fructose. Because the OH is on the right, you call it D-fructose. Clear? So this is configuration of monosaccharides or the D and L isomers. Clear? Isomer because they have the same molecular formula, same molecular weight, same everything. But their optical activity is different. This L-glucose will shift the plane polarized light to the left, while the D-glucose will shift it to the right. This much is clear? Okay, fine. One more thing I wanted to add is your body prefers D type of carbohydrates. Okay, so all the sugars, all the carbohydrates you find in your body will always be of D kind, D glucose, D glyceraldehyde. If somebody gives you L glucose, okay, just again, I'm just reminding you, think of glucon D, right? So why it is glucon D? The D stands for dextro, okay, D glucose. So if somebody gives you L-glucose, your body cannot metabolize this L type of isomer. Okay, that L type of isomer has OH coming out of some other side, and this cannot fit properly in the enzymes, as we shall see in the end of this unit, because of which your body will not be able to metabolize this glucose, and so your body will crave for the energy. Okay, it will not get energy. So all the sugar molecules in your body are of D type, dextro type. Clear? Fine. Now coming back to some basic chemistry. Here we are analyzing the reaction between two different functional groups. Just pay attention. Okay. You start with alcohol. alcohol here is shown as R2OH. This is the R chain. It could be anything, but the functional group is OH. And aldehyde. Aldehyde is CHO, and here also there is some R group. Fine. So when the aldehyde group and the alcohol group they react, they give you what is called hemiacetal. Fine. You can see the double bond between carbon and oxygen is broken, and that double bond is used to make a bond with this OH. So now what you get is this R1, then this middle C of CHO. With that C will be your OR2. Here from this oxygen, your carbon will get attached. So you have OR2. On the other side, this oxygen, which lost one double bond, will get a hydrogen from the alcohol and becomes OH. And here you already had an H. So this is called hemiacetal group. Okay, COH, then C has one H, and through one oxygen it has another R group. Okay, R chain. So this is called hemiacetal. Alcohol plus aldehyde gives you hemiacetal. Same way, alcohol plus ketone gives you hemiketal. Okay, ketone group is this C double bond O. It has R1 and R3. Okay, it has two side chains. And when the alcohol reacts with this carbonyl carbon, it gives you hemiketal. Clear till here? So just remember hemiacetal, hemiketal groups. Why is it important? Because the glucose in your body is rarely in this form. Okay, whatever we just studied, that is called Fischer projection. And the glucose in your body is not in this Fischer projection form. Okay, it is mostly in this cyclized form. It is in cyclic form. How does this cyclization take place? So the first carbon, which is your functional group carbon, okay, first carbon CHO, this CHO attacks the fifth carbon OH. Now what happens when aldehyde reacts with alcohol? You get ketal, acetal, right? So acetyl carbon, hemiacetal carbon. So this is what you're getting, okay? When the first carbon reacts with the fifth carbon, this is what happens. And you get this kind of structure where the sixth carbon will be outside the ring, and you have 1, 2, 3, 4, 5 membered ring with oxygen in between. Clear? So this is the cyclic monosaccharide. Again, OHH, OH. You can see the trend follows, and this is nothing but a glucose molecule, okay, in cyclic form. Now let us quickly see how to draw this cyclic form. You start with a vertical line, draw the corner oxygen atom, okay, so this is oxygen, and then you draw a hexagon. Now this along with this oxygen and the red line you see, and the carbon here, okay, this carbon, this red line, and the oxygen are showing you the hemiacetal carbon. Okay, it is showing you the functional group where that aldehyde group is present. It is present here. Then draw some vertical lines like this, number it. This will be carbon number 1, 2, 3, 4, 5, and the sixth carbon will be outside. Okay, the CH2OH, the last carbon will be outside. Now just place the OHH, OHOH, and the opposite ones, and here you have what is called alpha glucose. Okay, here you have a glucose molecule. This whole, what is shown in red is your hemiacetal functional group, and this hemiacetal functional group, if it has OH with a functional group, if the OH is present on bottom, it is called alpha glucose. If it is present on the top, OH, then it is called beta glucose. This is again a type of isomer, and these type of isomers are called alpha beta anomers. Okay, so here you have alpha beta anomers, alpha glucose and beta glucose. Remember this much, it will be important when we study disaccharides and polysaccharides. Okay, alpha beta. Beta means OH on the top, alpha means OH on the bottom. Now let us draw fructose. Fructose actually has ketone group on the second carbon. So the second carbon will attack the fifth carbon, okay, and will form a hemiketal group. This is called a furanose ring, okay, 5-membered ring. It's furanose ring. Here you have CH2OH and CH2OH. This will be the first carbon here and the sixth carbon here. Remember, fructose is also a hexose. So 1, 2, 3, 4, 5 and 6 carbons. Just same thing as you had written in glucose. Replace OH, OH everywhere. And here, because that OH is down, okay, it is at the bottom, you call it alpha fructose. If it is at the top, you call it beta fructose. Clear? So this is how the monosaccharides, they cyclize, they form this cyclic form, and they have anomers called alpha anomer and beta anomer. Right, so we just saw the structure of glucose in Fischer projection and in cyclic form. Now galactose, glucose and galactose are aldehyde aldohexoses, okay, which means they both have aldehyde group. So how are they different? So with the same molecular formula and almost the same structural formula, the only difference is on carbon number 4. You can see here on the fourth carbon, OH is on the right hand side in case of glucose, while it will be on the left hand side is in case of galactose. That is the only difference. And such kind of isomers are called epimers. Clear? So glucose and galactose are epimers of each other. They are different only at carbon number 4. Now that is all the only thing you have to remember, right? So if somebody asks you whether this is glucose or galactose, if you know glucose, you know it is OHH, OH. If on the fourth carbon the OH is present on the left hand side, you know that it will be a galactose sugar because glucose and galactose are epimers, okay? They differ only at the fourth carbon. Clear? When we studied the fructose structure, then here is the ribose, which is a pentose, okay, aldopentose sugar, which I hope by now you will be able to very easily write it down. Yeah. So here you have a ribose sugar, and when you cyclize the ribose, it gives you this kind of a structure. It is also having this 5-membered ring. Okay, the fifth carbon will be outside, and the 4 carbons forming the structure. If the second carbon has OH, it is the normal ribose. And we shall see in the DNA, the ribose sugar loses one oxygen. So here there is an anomaly, a difference. The carbon has 2 hydrogens attached to it. So this is called deoxyribose. One oxygen is missing. Clear? So ribose and deoxyribose. What is the importance of monosaccharides? They are building blocks of polysaccharides, disaccharides as well as oligosaccharides, right? So all the further sugar molecules or carbohydrate molecules are made up of monosaccharides. It plays an important part in energy metabolism, as you might have already understood because we discussed glucose and fructose. Ribose sugar is part of nucleotides. Okay, it forms DNA and RNA. And monosaccharides also store energy. Okay. Following, we study disaccharides. Di means 2. So here 2 sugar molecules. As you can see here, 2 monosaccharide sugars. They come together. When they come together, the OH and the OH, which are highlighted here, right? They are the functional groups. So the first carbon or the here what you see, the hemiacetyl carbon. That hemiacetyl carbon has OH. This OH will react with the fourth carbon here. This will be the fourth carbon's OH group. Okay. When that happens, H2O, one molecule of water is removed here. And there will be connection between this carbon and this carbon through an oxygen atom. This is called glycosidic bond or glycosidic linkage. Clear? Between carbon of one sugar and carbon of another sugar, The bond formed with a single oxygen atom is called glycosidic bond. The formula here will be CnH2O n minus 1 because one water molecule is removed, right? So CnH2O n minus 1. For example, sucrose, which is a disaccharide, right? Table sugar, the sugar you put in your tea, coffee, milk, that sugar. The formula is C12, okay, it has 12 carbons, so C12, H2O, 12 minus 1, because one molecule will be lost in formation of this glycosidic bond. So the formula will be C12H22O11, right? Seems familiar, okay, right? Fine. Examples are sucrose, which is a table sugar, okay? It is made up of two monomers or two monosaccharides called fructose and glucose. Okay, you can see the structure here. This is your glucose, and just for information, okay, no ratta marofine this, but the scientific names are also given here. This is alpha type of sugar, which means on this first carbon the OH will be present at the top, okay, above OH. Then D, glucopyranosyl. Okay, I told you this is a pyranose ring. So this is the scientific name of glucose, or what kind of glucose is present. So alpha D glucopyranosyl. Then where the bond is forming between the first carbon of glucose and second carbon of fructose. So you call it 1, 2. Okay, this 1 and arrow 2 is showing the bond between the two carbons, which is nothing but the glycosidic bond. And then beta fructofuranose. This ring, which is formed because of ketone reacting with alcohol, is called furanose ring. And this furanose ring is of beta type, okay, which means the OH will be present at the bottom. So this 1 and 2, these carbons, they are forming a glycosidic bond. Okay, here is the oxygen atom. So this is how sucrose molecule is made. Okay, it has one glucose and one fructose. Then you have lactose, which is milk sugar. It is made up of galactose plus glucose. Now, what I suggest to my students is to remember galactose first and glucose later, because that is how the molecule is forming. You have galactose coming first, followed by the glucose, and there is a bond between them. That bond is 1,4 type. Okay, the first carbon of galactose and the fourth carbon of glucose will form a bond, and this is what is giving you lactose. Finally, there is maltose, which is the grain sugar. Okay, when you eat, let us say, a piece of chapati, you chew it for a long time. The sweetness you get from chapati is because of maltose and glucose. Okay, it is basically a disaccharide made up of glucose and glucose. So here you have two molecules of glucose which are attached to each other through a glycosidic bond between first and the fourth carbon of the sugars. Clear? Fine. Now let us just quickly discuss what is reducing and what is non-reducing type of sugar. In reducing sugar, you have a free aldehyde or hemiacetal or a ketone group or hemiketal group. You have some free functional group. Free in the sense it is able to react. Just take the example shown here, the diagram shown here. Can you see in the first sugar, the hemiacetal group is involved in formation of a bond, which means it is no longer active. It will not be able to react. But the hemiacetal group of the second carbon, shown here in red, is not forming a bond. It is open. It is active, right? So it can reduce. Reduce means to add hydrogen to some other molecule, right? So this here can reduce other molecules. It has ability to reduce cupric, which is Cu2 plus, or Fehling's or Benedict solution to cuprous, which is Cu1 plus. Okay, so from Cu2 plus to Cu plus, this conversion, if the sugar is making, you come to know that it is a reducing type of sugar. Example are glucose, maltose, lactose. Okay, all of these are reducing sugars. What are non-reducing sugars? Here, the sugar does not have any pre-functional group. Okay, pre-functional group is not present. It is involved in a bond because it is not present. Okay, free, so it will not be able to reduce cuprous, cup, cupric to cuprous. Example is sucrose and trehalose. Just see here, you have glucose plus fructose. This glucose and fructose are forming a bond between the hemiacetal and the hemiketal groups, which means no functional group is left free for reacting. So this is a very non-reactive, very stable, and very non-reducing type of sugar, sucrose. Yeah. So just go and read up about this, revise about this. From the next time, we will be starting with some more things about carbohydrates, especially polysaccharides, followed by nucleic acids. Okay. See you next time.